Hello everyone, welcome back. Today we'll be trying to take a topic which is very common for all the science students, right from class 11 to the graduating third year students. So the today's topic is from biological chemistry and it's glycolysis, the first stage in cellular respiration. So over here we'll not be trying to take it in way how you have studied it in biology. Rather than we'll be trying to understand the chemical reaction, how the structure changes takes place, how the groups are transferred from one position to the another, how the isomerization takes place, how the phosphorylation takes place, how the oxidation takes place. So all this chemical reaction will try to understand, and what are the various enzyme involved. So that is how we'll be taking glycolysis in this particular video. So, before going into the chemical reaction, let us try to understand what glycolysis is all about. So, glycolysis is a term used to describe the sequential series of reaction. As I have told you, we'll be discussing those reactions present in wide variety of tissues that starts with a hexose sugar. So, what do you mean by hexose sugar? It is that carbohydrate which contains 6 number of carbon atoms. Remember that carbohydrates are classified into different categories, and depending upon the number of carbon atom which they possess, they are classified into different categories, and the one which contains 6 number of carbon atom is known as a hexose. In our case, usually glucose, and ends in pyruvic acid. So glycolysis is nothing but a process where a glucose converts to pyruvic acid through a various sequence of enzymatic reactions, and those enzymatic reaction will be trying to understand. So what do we mean by enzymatic reaction? Those reactions which are catalyzed by enzyme. And one important thing, remember that glycolysis takes place in cytosol. What is a cytosol? A cytosol is the aqueous component of the cytoplasm of a cell, okay, which contains various organelles, cell organelles, and the particles suspended in it. Okay. Now, glycolysis is the first stage in the breakdown of glucose, and it's common to all the organisms. So when we say that, so it means that it is common to both aerobic and anaerobic modes of respiration. Now, glycolysis is originated from two Greek word. One is glykos and another one is lysis. Glykos means sugar and lysis means splitting. So basically it is breakdown of sugar molecule or glucose molecule in our case. The scheme of glycolysis was discovered by three scientists, okay, Gustav Embden, Otto Meyerhoff, and J. Parnas, and therefore referred to as EMP pathway from their abbreviation of their last name, okay, Embden E, Meyerhoff M, and Parnas P, okay, so that is why we call it EMP pathway. Now, the steps of glycolytic pathway. What are the various steps involved in glycolysis? So let us try to understand one step at a time. So the first step. So the first thing what happens in glycolysis is phosphorylation of sugar. Now what do you mean by phosphorylation? It is that reaction where a phosphate group is added. Okay. Now in case it is of sugar, so it will be added to a sugar, and remember that derived from ATP. Okay. So the sugar which we are talking about is glucose. So the glucose, or this particular step, involves the formation of glucose 6-phosphate by a hexokinase. That is the catalyst. Hexokinase catalyzed phosphorylation, as I have told you, of D-glucose with ATP. So when D-glucose is phosphorylated with ATP, what we get is glucose 6-phosphate, which you can clearly see. The left hand side molecule is the glucose. The glucose reacts with ATP, and you can see the CH2OH, the sixth carbon of glucose, okay, CH2OH. That CH2OH. Now in the right hand side, glucose 6 phosphate, what you can see is it's not CH2OH, it is CH2O and PO3 2 minus, that is phosphate group. So a phosphate is introduced in glucose in 6th position. That's why we call it glucose 6 phosphate. And from where we are getting that phosphate, you can clearly see ATP in the reactant is converting to ADP in the product. That means adenosine triphosphate, 3-phosphate, now it's converted to adenosine diphosphate, 2-phosphate. One of the phosphate is transferred to glucose, which converts it into glucose 6-phosphate. This process is known as phosphorylation of sugar or glucose. Now, once the glucose 6-phosphate forms, now the next step is the isomerization of glucose 6-phosphate. So what basically we do is we isomerize the glucose 6-phosphate to fructose 6-phosphate, and that is done by a catalyst, phosphoglucoisomerase, which catalyze, as I have told you, the isomerization. Isomerization is a process of converting one isomer to another, that is of glucose 6-phosphate into fructose 6-phosphate. Now if you see the basic difference between this, you can clearly see that a glucose 6-phosphate is converting into fructose 6-phosphate, and remember that a 6-membered glucose is converting into a 5-membered fructose. Okay, so that is what happens. So that is the isomerization of glucose 6-phosphate into fructose 6-phosphate. Okay, in this particular reaction, a 6-membered ring converts to a 5-membered ring. Okay, or we can say a aldose. A glucose contains a aldehyde group, so we call an aldose, converts into a ketose. Okay, into a fructose, where actually fructose contain a ketonic group, so it is a conversion of an aldose to ketose. Okay, then after the formation of fructose 6-phosphate, again phosphorylation takes place. Now I don't have to tell you what is phosphorylation. It is nothing but the addition of phosphate. So we add one more phosphate group in fructose, so that is done by phosphofructokinase catalyze the phosphorylation of second primary alcoholic group of fructose 6-phosphate with a second molecule of ATP, forming fructose 1,6-diphosphate. So you can clearly see in the left hand side I have fructose 6-phosphate, and in fructose 6-phosphate I hope everyone can see CH2OH, the first carbon. Okay, that CH2OH is a primary alcoholic group. Okay, OH is alcoholic group, it's a primary alcoholic group, CH2OH, and that CH2OH is phosphorylated with ATP again. Okay, giving fructose 1,6-diphosphate. Now you can clearly see in the 6th position of fructose you have a phosphate group. Now in CH2OH of fructose 6-phosphate, the primary alcoholic group, you can see in the product, in that CH2OH, in place of CH2OH what you have is CH2O PO3 2 minus. Okay, once the fructose 1,6 diphosphate forms, then it undergoes aldolase catalyzed fragmentation. It basically split, that's why the step is known as splitting. So it splits or it fragments into two molecules, that is glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Now a point to be noted over here, it forms two molecules, but remember that the desired product is glyceraldehyde 3-phosphate, not dihydroxyacetone phosphate. So glyceraldehyde 3-phosphate will be the desired product and not dihydroxyacetone. Okay, so you can clearly see the fructose 1,6-diphosphate now converts into two molecules, one of glyceraldehyde phosphate and another of dihydroxyacetone phosphate. So that means you get 1 mole of glyceraldehyde 3-phosphate and another mole of dihydroxyacetone phosphate. Now as I have told you that the dihydroxyacetone phosphate is not the desired product, so in the next step what we do is we do isomerization of dihydroxyacetone phosphate to glyceraldehyde 3-phosphate. So again, we'll be going to do isomerization in this process, where we use a catalyst, triose phosphate isomerase, where the dihydroxyacetone phosphate, as I have told you, not the desired product, converts into glyceraldehyde 3-phosphate. So all the molecule of glucose is finally converted into glyceraldehyde 3-phosphate. And remember that 1 mole of glyceraldehyde 3-phosphate forms in step 4, and the another mole, which is actually the conversion of dihydroxyacetone to glyceraldehyde 3-phosphate again. So in step 5, again you will get glyceraldehyde 3-phosphate. So in all, you get 2 molecules or 2 moles of glyceraldehyde 3-phosphate. So that is how the isomerization takes place. The dihydroxyacetone phosphate converts to the glyceraldehyde 3-phosphate. Once the glyceraldehyde 3-phosphate forms, then the glycolytic pathway enters the energy conserving phase. Here it is oxidized. That means the glyceraldehyde 3-phosphate is oxidized to 1,3 biphosphoglycerate. Okay, so we add or we convert into biphosphoglycerate by NAD, that is nicotinamide adenine dinucleotide. This reaction is catalyzed by an enzyme, glyceraldehyde 3-phosphate dehydrogenase. So what happens? Glyceraldehyde 3-phosphate is oxidized by NAD, and remember that in presence of inorganic phosphate, HPO42-, inorganic phosphate, which gives you 1,3 diphosphoglycerate. So the difference you can clearly see. In case of glyceraldehyde 3-phosphate, you have a phosphate group in the third position, PO32-, in the third carbon. In case of 1,3 diphosphoglycerate, you can clearly see you have 2 phosphate group, one in the first carbon and another one in the third carbon, and with the release of NADH and H+. Once the formation of 1,3 diphosphoglycerate takes place, then the formation of ATP takes place. So what happens now in this step is the 1,3 diphosphoglycerate or biphosphoglycerate transfer one of the phosphate to ADP now. So the ADP takes one of the phosphate, the 2-phosphate which is present in biphosphoglycerate, one of them is taken by ADP to convert to ATP. And this happens in the presence of phosphoglycerol kinase. Okay, and the 1,3 biphosphoglycerate converts to 3-phosphoglycerate. So you can see in the left hand side 1,3 diphosphoglycerate, 2 phosphate group is there, PO32- in the first and the third position, reacts with ADP, adenosine diphosphate, transfer one of the phosphate to ADP to convert into ATP, you can see in the product, and one of the phosphate group in 3-phosphoglycerate. In the product, you can clearly see 3-phosphoglycerate. The first carbon does not have phosphate anymore. It converts to a carboxylate, COO-, or we can say carboxylic acid. Okay, and one of the phosphate remains in 3-phosphoglycerate. Okay, in the third position. Then the next step is the isomerization of 3-phosphoglycerate to 2-phosphoglycerate, and that is done by an isomer phosphoglyceromutase. Okay, so what we do is we convert 3-phosphoglycerate to 2-phosphoglycerate. You can clearly see in 3-phosphoglycerate in the left, you can see the phosphate is in third position. Then in case of 2-phosphoglycerate, it goes into the second position. Okay, so remember that it's just a isomerization process. Once the 2-phosphoglycerate forms, then the dehydration of 2-phosphoglycerate takes place. Okay, by dehydration, what do we mean? Dehydration means removal of water. So we remove water from 2-phosphoglycerate by using a catalyst, which is nothing but enolase. Okay, the enolase catalyzes the dehydration of 2-phosphoglycerate, producing phosphonyl pyruvate. So you can clearly see in 2-phosphoglycerate in the left hand side, there is CH2OH. OH is in the third carbon and H is in the second carbon. The OH and H, the OH from the third carbon and H from the second carbon removes as H2O, and what gets introduced is a double bond. and the product which you get is known as phosphonyl pyruvate, okay, having a phosphate group in the second position. Okay, now in the final step of the glycolysis, the phosphate group which is present in phosphonyl pyruvate, as we can see, is transferred again to ADP, okay, converting it into ATP. So this is the final step of glycolysis. This step actually is catalyzed by pyruvic kinase, where the phosphonyl pyruvate actually donates its or transfer its phosphate to the ADP, producing ATP, and phosphonyl pyruvate changes to pyruvate. That is our the final product, or pyruvic acid. So the last remaining phosphate is again transferred to ADP and giving you ATP, and the final product of glycolysis, that is pyruvate or pyruvic acid. So the point to be noted over here is, so if you try to understand the overall process of glycolysis, in this particular process, 1 mole of glucose, remember that, converts into 2 moles of pyruvic acid, okay, and the 2 moles of ADP get phosphorylated to form 2 moles of energy rich ATP. So ultimately you get 2 moles of ATP in glycolysis. So the overall step can be understood in this way. Okay, so that is the overall step of glycolysis. These are all the steps through which a glucose converts to a pyruvate. And the point to be noted is that at the end you get 2 moles of energy rich ATP in glycolysis. Thank you everyone. Do watch the video uploaded in this channel and subscribe for the channel for the explanation on various topics in chemistry. Thank you very much.